Hi, everyone. Welcome to our podcast, The Kids Cancer Therapy Toolbox. Today, I'll be interviewing Alyssa Almer, a former cc-TDI intern and immunotherapy specialist at Genentech. So, to start us off, could you explain, for those who aren't familiar, what the role of an immunotherapy specialist is?
Yeah, of course. Hi, Alyssa. I work at Genentech as an immunotherapy specialist, and what my role is I work in cell and gene therapy clinical manufacturing. So, immunotherapy specialist is kind of a fancy title, but we work to help develop and manufacture cell and gene therapies that are targeted and curative. So things like CAR T cell therapies or stem cell therapies, they're very individualized as well.
Okay. So I understand you have an interest in engineering. How did that lead you to this job and your past internship with CCTDI? Because I know they're a bit different.
Yeah, they are a bit different, definitely. cc-TDI is a little bit more, R&D, but I've always been interested in science and math, and bioengineering specifically really combines those in a very unique way that kind of brings that, like, abstract math to being able to make curative therapies. So
Nice.
cc-TDI, I actually heard about it from my neighbor who was previously an intern there, and then I went to an open house, at cc-TDI when I was in my freshman, sophomore year of college, and the kinda, like, the message really spoke to me. My childhood best friend had leukemia in high school, and then in college, I was working in a bioengineering lab creating 3D in vitro models of different cancers. So when I went to the open house, I kind of saw an opportunity and to learn and combine the engineering with, the biology of it.
That's amazing work. What would you say are some of the advantages of using computers rather than other models, such as animals?
Yeah, so there's multiple different advantages of using computer simulations or in Cisco trials rather than using mouse models or primate animals for testing. So for computer models, it is a lot faster. There's a lot less time and money constrictions on being able to make a computer model. It's also extremely patient specific. So you can design it based on different patient parameters that you get from normal testing, MRI scans, CT scans, and the results are also a lot faster. You don't have to wait for the animal to uh, either grow the cancer or wait for to see what the reaction is. You can get results pretty instantaneously. It's also a lot more accurate. So if you're comparing, how something's going to react in a mouse compared to a human, the reactions and the pathways are very different. And so if you can use a computer model to say this is exactly or this is how we expect human parameters to react, then you can get something that's a lot more accurate.
Would you say that's the future of cancer treatment development more than like moving away from animal models?
I think that's definitely the future. I think using a combination of the computer simulations along with AI, which that's kind of everywhere now,
Hmm.
um, it's gonna allow for a lot of research, a lot of the R&D to be sped up. And that is kind of what takes up a lot of time when you're trying to get medicine and therapies to
Yeah.
patients, either clinical or FDA approved. It's really that R&D that takes a lot of time to get results, and also um, it takes a lot of time to get to a point to where you want to use an animal model. And once you get there, a lot of the um, discovery drugs or therapies, that's where majority of them fail. And so if you
Mhm.
can find something to bridge that gap of, testing something on a petri dish or on like a plate, just on a 2D surface, and getting that somewhere closer to how it could look in a human or a patient, you really bridge that gap and you speed up the process of finding out what is going to cure people.
That's amazing.
It's really cool place to be. Yeah.
Yeah, yeah. Are there any more kind of common applications? I know you talked a bit about, like, just how the computational biology plays a role in childhood cancer research, but, is there anything more you'd like to say on that?
Yeah, so computational models like for biology can be used not just for kind of the project that I worked on at cc-TDI, which was the fluid dynamics, but it can also be used to analyze really large sets of data, and that can be done. Um, it can be done by using large sets of data to figure out what type of treatment um, will best fit a specific patient. So a child could have the same type of cancer, but they're going to react very differently to the same type of treatment. And so computational biology can allow you to take a bunch of information from databases and from that patient, DNA, RNA, and specifically target what will best suit their biological needs. Um, it's also it's also really good at being able to predict the pathways and the pharmacokinetics of how a drug is going to react in a body before you move it something into a clinical trial.
Okay, so multifunction.
Yeah.
Yeah. What would you say, are some of the advantages of being an engineering major working in biology? Does that I imagine that plays a big role with the computational biology, but is there anything else specific?
Um, I think that being an engineering major, you're able to take the visualization of, okay, you know how the body works, you know how the cells are going to work,
Mhm,
and being able to have that engineering math, you can kind of what we previously talked about. You can take that and you can model it. You have a better understanding of not only how the body works, but why it's working in certain ways. Um, and I think that bioengineering, chemical engineering, having that engineering, the math, the science being integrated into the biology, just has an added benefit of being able to piece together all the little puzzle pieces.
mhm.
And I think you're able to see more of a bigger picture, and not only the how, but also the why. And then engineering is also you're creating something. So basically, bringing science to life in a way.
Yeah, that sounds really cool. Um, so now I'd like to move into a little discussion of your recently published paper. I think it was in May. Yes.
Yeah.
Okay, okay. Would you mind giving a brief summary of the project for those who aren't familiar?
Yeah. So this project is one that I worked on while I was an intern at cc-TDI, and then I continued to work on it when I went back to college. But there were also a lot of other collaborators and authors who helped on this. But it was a project that combined patient-specific computer simulations with lab experiments in order to understand how to treat medulloblastoma that's spread on the surface, and we specifically looked at a drug called BMS. Not sure if you want the name, but we looked at an investigational drug to target the IGF-1 receptor to see how that would look when you are spreading it through cerebrospinal fluid.
Okay. What exactly is medulloblastoma and why is it so unique?
Yeah. So medulloblastoma is one of the most common malignant brain tumors in children. Brain cancer in general is one of the most deadly brain cancers, but in children, it's unique because it has a high tendency to metastasize. So medulloblastoma will spread through the CSF, which will then go down into the lining of the brain and the spinal cord, and that's really what makes it unique, and specifically difficult to treat with traditional chemotherapy or radiation, is because it does spread through the cerebral spinal fluid.
Mm. How did you create a model for that spread, and then the delivery of the drugs?
Yeah, so we worked with Bryn Martin, who he was really helpful on the engineering side and helping create the computational fluid dynamic model. So we took a patient's MRI scans, and from those scans, we were able to outline where the fluid flows in the brain, so between the brain and the spinal cord. And then we used engineering software to simulate the 3D movement of the CSF. And after we were able to model it, we modeled how the drug would be injected into the brain, and the computer was able to model the spread based on the flows that we put into it.
Did your math background play a role in the development of the models, or how did math in general play into your project?
Yeah, so the modeling of how the fluid actually flows, that is all engineering equations. So if you anyone who's an engineer out there, Navier-Stokes equation, continuity, those are all very basic fluid dynamic equations that most anyone with an engineering background will learn or come across at some point. And so those equations are model any type of fluid movement based on different parameters, and that can be
Mhm.
modeling anything from the flow of a faucet to cerebrospinal fluid. And so that it's essentially the engineering and the math. You're taking what you see and you're putting it into numbers and variables that you can then adjust based on patient specific information.
Mhm.
Let me know
So
if you want a little more detail on that or not.
I think you explained it perfectly. I'm just kind of wondering how many engineering majors there seems like there's so many different directions to choose from because you have these formulas that can be used for like a wide application of things. Do Would you say a lot of engineering majors, or how big of a percentage of engineering majors are choosing to put their talents into biology?
I think that really depends on um, the type of engineering they decide in school or their passions.
Mhm,
So you have dozens of different types of engineering majors: chemical engineering, bioengineering, mechanical engineering, industrial engineering. So I think that I'm not sure I can put a percentage on how many engineers
oh,
are
yeah,
putting
that's
their
fine.
But, I think that majority of people who go into bioengineering or chemical engineering, biomedical engineering, that is really what their focus is on. It's either gonna be something on devices or on, drug or medicine treatment development, and
Mhm,
so that at least specifically the program at Oregon State that I went to and having the opportunity at cc-TDI really allowed me to see how I could directly translate what I, learned in class to, functional applications. And so I think that definitely the cc-TDI internship had a big impact, on me and how I was able to kind of see where I wanted my career, to go.
yeah. Um, Circling
That answered
back a
your
bit.
question.
Yes, it totally did. Circling back to the paper a bit, how did you confirm that the model you had made was accurate?
Yeah, so the model's accuracy was verified by checking numerical timestamps and mesh independence. So mesh independence is kind of how the drug and the fluid flows kind of over time. So we compared that with previously published experimental data on how different molecules um, trans transport through the human CSF. So we used the data that we got from the patient. We used other published data from similar molecules, and then we used we used that in combination with the output from the model in order to see how accurate like the fluid velocity was flowing or the pressure. And then based on that, it was adjusted.
Okay. How were the models able to accommodate the patient-to-patient variability and other factors?
Yeah, yeah. So for this specific model, it was designed for um, kind of one patient, and that was designed based off of the MRI scan. So once we had that base model, we were able to take a specific patient's MRI data, which had like stroke volumes and flow patterns and pressures. We took that and we put it into the model, and it adjusted itself. And you can do that with any other patient. This wasn't a um, anything super unique to the to getting the scans from the patient. And
Okay.
so once you kind of have the base model, you can any patient that wants to go in to do an MRI scan, there are certain parameters that you can get from those, and so how you would adjust any other kind of algorithm or equation, you just need to put those parameters um, into that computational model.
Okay, thanks for clarifying. Um, just
Yeah.
one last kind of general question. Where will you go now, and how did your internship lead you to your career selection, work at Genentech, and just where you are today?
Yeah, so I think cc-TDI really gave me a foundation for what I am passionate about in the intersection of engineering and science/biology. So after cc-TDI, I continued to work at the bioengineering lab at Oregon State, where I actually, for 3 years, I worked on a project that had a very similar goal.
Mhm,
Um, at Oregon State, we were using technology to create 3D in vitro models of how cancers react to drugs by mimicking the microenvironment. So we were also trying to kind of bypass um, the animal models in a way and find something that is a lot more accurate and quicker at mimicking what happens in a body. Um, that this was a little bit less technology, a little bit less engineering that I used, but kind of from cc-TDI and from the lab at Oregon State, um, I then got an internship at Genentech, which is now where I work. And in Genentech, it's a bit different. It's a little bit less R&D. It's more process development, but I'm able to use the engineering and then the biology to help push forward the understanding of getting um, developing the best process to get product to patient.
mhm.
So I think that cc-TDI really laid a foundation for my passion of speeding up the um, the R&D process in a way that is viable. So not necessarily speeding it up to just, push it along, but actually being able to speed it up in a way that we are advancing the technology, getting more better and accurate more information, and getting it out into clinical trials faster. So yeah.
It sounds like you're doing amazing work. Thank you so much for coming on and interviewing today. We're very grateful.
Yeah, thank you so much for having me.